Now, DNA polymerase has some issues, okay? Chief among them, it has to have something to build on. That 3' carbon, okay? Hence, you have to have primase, laying down RNA primers. Well, what happens if you run out? You hit the end of a linear chromosome. There's no place to lay down a new primer. So there's no way to lay down and finish all of the lagging strand. What you would end up with is a strand that gets shortened. A strand that gets shortened here, another strand shortened there. Next round of mitosis, it could be the same chromosomes again, it could be different chromosomes, and it just goes on and on and on, and this could be a problem. Because how much could you lose before you start getting into the genes? A gene that is important for fetal development, a gene that is important for maintaining a cellular metabolism. Which is why eukaryotic chromosomes, since they are linear, have evolved to have what are called telomeres. Telomeres are the ends of the chromosomes. Within the telomeres, there can be genes that are used once and never again during early fetal development, or they're gobbledygook DNA, buffer DNA, so that as the telomere gets shorter and shorter after so many rounds of mitosis and DNA, and cell division, that all that is lost is junk at the ends while protecting the important genes within the center of the chromosomes. Now, the telomeres don't prevent the shortening of the DNA molecules. This is going to happen on its own. They're going to shorten every round. You're going to lose a few dozen nucleotides. But what it does is it slows down and allows for, round and round and round of mitosis before you start to get into genetic material, important genetic material. There are some avenues of research that are saying that the telomeres are actually the cell's clocks. That as the telomeres shorten, the cell will age. It's how our body knows, adolescence, teenage years, which means the onset of puberty, post-puberty, you get your, fertile years, then the onset of, the loss of, sex hormones as we age and things like that. How does our body know where we're at in the temporal time frame of our life cycle? They're assuming it's telomeres and the overall telomere lengths. Every chromosome has telomeres at each end. There are enzymes known as telomerases or telomerases that will go and will randomly add in nucleotides at the end of the telomeres. So you lose a couple dozen and you'll gain back a dozen or so. Lose some, gain back some. Not complete, not all, but some. They've shown in, I believe it's mouse models, that when you have an overproduction of telomerases, which means the telomeres get lengthened back to almost normal, original length, that it will prolong the life of the mouse. So they're starting to think this may be our humans' fountain of youth. We need the telomeres there to protect, because as the telomeres shorten, you want to make sure that there is a buffer before you get to the genes. But you want to allow for the telomeres to shorten because it will qualify the lifespan of the cell. Because a cell that's near immortal, that keeps being able to reproduce, will never time out, and over time could begin to accumulate mutations. So by having a lifespan, any mutations that have accumulated will die off with that cell. Also been shown that in many cancer cells, telomerase activity is lost. So the chromosomes shorten and they begin to lose whatever genes are at the end of the chromosomes. So we'll see what the future holds for the study of telomeres.